Good morning to all of you. Myself Amol Borade, host of today's session. I welcome you all for today's second pharmacovigilance lecture. First of all, I welcome Dr. Mohan Kale sir on this second pharmacovigilance platform. Welcome, sir. For today's lecture, we are very fortunate to have two eminent speakers with us from the industry. Dr. Ramandeep Singh Bedi sir and Dr. Manpreet Kaur madam. I welcome both the guests for today's session.
Thanks, Amol.
Thank you. Good morning.
Thank you, sir. Thank you, ma'am. For our first lecture, I would give the brief introduction of Dr. Bedi sir. Dr. Bedi sir is currently working with Mylan Laboratories, Bangalore. Currently having the position as Associate Director in Formulation Development. He is having near about 13 years of experience in the industry. He has also worked with Panacea Biotech and also with Signet Chemicals, Mumbai. In his credit, he is having many numbers of research as well as review article, and he has also attended various international and national conferences. So with this brief introduction, I would request Dr. Bedi sir to take the today's session.
Thanks, Amol, for that brief introduction. So basically, I'll be just giving you a brief about pharmacovigilance and its relevance. You all know these days pharmacovigilance, especially in common people, it is very much there. Like when we are talking about vaccines, whether these are safe or not, what are its efficacies and all, that all depends how the media is percolating the message. So right now we are vigilant, whether we should take the vaccine or not. So that is nothing but pharmacovigilance. There is a special team in industry, pharmaceutical industry, which deals with all these issues. So I'll be just briefing you what exactly it is. So first of all, I would like to tell how formulation development is being carried out in industry. So there is a preclinical research. So when we are talking about vaccines or we are talking about formulation, so first we do a preclinical research where we do synthesis and purification of a material. Then we do animal testing, and then it can be short term just to check how acute and chronic toxicity of that molecule works. Along with that, long-term studies in which various doses are various levels are being given. Then the formulation is being developed and IND is being submitted. When the IND is submitted, that goes under various clinical reviews studies. First is phase 1. We all know that when the vaccines are being produced, we were all very excited. Okay, what is happening? So there was a phase 1 study. So what does phase 1 study mean? Phase 1 study means that we have to dose the vaccine or the formulation in animals. If the animal respond to that, if we get a toxicity established in animals, afterwards we go for phase 2 trials. In phase 2 trials, we do it on limited number of patients, maybe around hundreds. In those 100 patients, we generally establish what is the safety of that formulation, not the efficacy, but the safety. How safe is the formulation? So that's why when Covishield and Covaxin was being developed, a lot of hype was there whether it is having any reactions or not. So that was determined during phase 2 trials. And then phase 3. Phase 3 will always be on the higher side, like it will be from thousands to maybe 10,000s or maybe million also. So that all depends how that formulation is going to work. Like Covaxin or Covishield, when we talk about, it was tested on more than 10,000 people to establish whether it is really efficacious or not. So phase 1 determines is determined in animals, where the safety and efficacy of the formulation is being established. Phase 2, where it is done on only a limited number of people just to check whether it is safe enough. Phase 3, where we check the efficacy in human beings above 25,000 or above 10,000. And afterwards, then the formulation goes for NDA review. And only after the NDA review, the formulation is told to get marketed. So the FDA estimates like 11 years the time span is for the entire process of preclinical research to NDA, and that is why a lot of people are having doubts whether the vaccines which are being developed are safe or not. But let me tell you one thing very frankly, that these vaccines are being developed from the past 1.5 years, and a lot of studies, like all these studies, like which are to be completed within 11 years. Those studies were completed within a span of 1 year to establish the efficacy and safety of the vaccines. So currently, one molecule, when one molecule is having a shelf life, then first 1 billion, approximately $1 billion is being spent on those formulations to get into the market. So you just imagine, out of 1,000 molecules, only one molecules go into the market because of various reasons. It can be safety, it can be efficacy and all. So you can just imagine near about $1,000 billion is being pumped when one single formulation comes into the market. So what is pharmacovigilance? Pharmacovigilance is nothing but vigilance from a person. So the science and activities related to detection, assessment, understanding, and prevention of adverse events or any other medicine related to the problem. That is being coined by WHO. So we have to assess whether the formulation is going to prevent the disease and what kind of adverse effect this is going to occur in the people. That is what right now pharmacovigilance is. So what do you mean by vigilance? Vigilance is being termed as vigilare, which is an Italian term which means to watch. So to watch means alert, watchfulness, like we are very much watchful right now, whether we should take the vaccines or not. Forbearance of sleep, that is wakefulness, we are alert all the time. Watchfulness in respect of danger, care, caution, and circumspection. The process of paying close and continuous attention. We all were very keen on knowing whether the vaccines or the tablets which we are taking during COVID are going to affect, prove the efficacy in the COVID or not. What is an adverse event? An unwanted or harmful reaction experienced following the administration of a drug, combination of drug under normal condition of use, and suspected to be related to the drug. We all know, I'm just taking the live example of the vaccines right now, because we all are very experienced in and we are having quite close watch on those vaccines being developed, and we all know what exactly it means. So when we say adverse events, like we are very much curious to know whether the vaccines which are being developed, are they having any adverse event or not, because then only we'll be taking the vaccine. But let me tell you, adverse events are different than side effects. Side effects is generally coined as a term which generally come along with the vaccine, and those are or the formulation, and those are dependent on the formulations, but it's not going to harm the product, harm the person as such. Adverse event is the one which will cause reactions to happen, and it may cause death also. So what are the common ADRs which we generally see? Like when we use codeine, then constipation occurs. When we use antihistamines, sedation occurs. When we use fluoxetine, nausea occurs. Similarly, when we use NSAIDs, when we use aceclofenac, diclofenac, we know that GIT upset is going to happen. So these are very common adverse drug reactions which are happening. We all know the classic case of thalidomide, in which thalidomide was given to pregnant females for treating nausea and vomiting. In the late 1960s, this was a common drug which was used to treat nausea and vomiting in pregnant ladies. So nobody knew what are going to be the side effects of this. So what side effects was known after that is people were giving birth to the babies who were having deformed limbs. So that was very much prevalent, and afterwards thalidomide was banned from the entire world. So medicines are supposed to save lives. Those are not supposed to take lives or cause deformities in the patients or cause adverse events to the patients. So as per WHO Geneva 2005, it was published that dying from a disease is sometimes unavoidable. Dying from a medicine is unacceptable. So we have to take care that the medicines which we are taking are for treatment, and those are not for causing adverse reactions or to cause deaths in patients. So what are the drug outcomes? When we give or deliver a dose to a patient, it causes some effects, which can be pharmacological effect or it can be therapeutic effect. Like when I'm taking paracetamol, it has to treat my fever. So that is a therapeutic effect. Similarly, side effects, which are unintended. We don't want those side effects to happen. But sometimes side effects do happen, like when we take the antihistamines. So these are going to have your GI upset. So this is a side effect. Interaction can be expected, interactions can be unexpected. Like we cannot take two simultaneous medicines in one go because it might have an interaction. So that's why most of the medicines which we are taking, in their pack inserts, it is strictly written that these medicines are not to be taken with these medicines. Like doxorubicin injection is not to be taken with bortezomib injection because it may cause thrombocytosis to happen. So in that way, interactions can be expected. These are the expected interactions, but unexpected, we don't know. Some people are having idiosyncrasies, like if the medicine is to be taken with milk, they cannot take it because it will have interaction. Some people are allergic to milk. Similarly, impact, whether it is beneficial or harmful. We all want that medicine should be beneficial to us, but it doesn't happen all the time. So it can be harmful also. So what are harmful effects? Mortality. Definitely mortality is the most harmful effect which we can ever see in a person, means the person dies. Along with that, we all know this term morbidity. Morbidity is nothing but having other diseases along with the disease which is currently in hand. We all know that diabetes patients or the heart patients right now having COVID, those are comorbidities. Like diabetes and heart patients are told if they are going through COVID, then they should take great care because COVID is going to impact diabetic and cardiovascular patients also. Similarly, harmful effects can lead to hospitalization, prolonged stay, extra treatment. Definitely extra treatment comes with the cost, and along with that, resources which are being consumed. So why we need PVG? Why we need pharmacovigilance? Because I've already told that we run phase 1 clinical trial, phase 2 clinical trial, phase 3 clinical trial. So when we are doing phase 1 clinical trial, then we are having very limited animal experiments, and those are very limited in predicting the human safety. So we do clinical trials. Clinical trials normally, the phase 2 clinical trial only runs for 100 of patients. So those 100 of patients are under strict surveillance, which are in hospital or which is with the medical reviewer. Along with that, the companies are also following up. And when we do the clinical trials, we generally don't consider patients who are having comorbidities or the persons who are less than, like clinical trials are generally conducted on healthy human beings ranging from 18 years to 55 years. So any person who is having age less than 18 or any person who is having age more than 55 will not be considered in the clinical trial. But we don't know when the patients are being administered, being dosed with the medicines, whether those populations like below 18 years and above 55 years are going to have any side effects or not. Or we generally don't take pregnant ladies during the clinical trials. We don't know whether those medicines are going to impact the pregnant ladies or not. So we all have discussed right now the classic case of thalidomide. So that was generally launched in the market after giving clinical trials to age 18 to 55. So they have not considered the pregnant females. So these are the shortcomings. So that's why we need pharmacovigilance. So what I told earlier, like we are not considering children, we are not considering the elderly patients, we are not considering the pregnant or lactating women, patients with comorbidities like hepatic or renal disorders or diabetics or heart issues are not being considered during the clinical trials. Similarly, subpopulations carrying known and relevant genetic polymorphisms. Similarly, patients of different racial or ethnic origin, like when we do a clinical trial, that is very limited. If we are doing it in India, it will be only on the Indian population. If we are doing in US, it will be only in US population. But how it is going to impact the African population, or we don't know how it is going to impact the Japanese population, because all are having different pharmacokinetics, all are having different build-up, all are having different body mechanisms. So that is why pharmacovigilance is very much necessary. How the drug development is happening, I'll talk about that right now. So the clinical development of medicines, how it is happening is we first do the preclinical animal experiments, which I've already told, where we check what is the acute toxicity, how organ damage is happening, dose dependency is there or not. It can be linear, it can be non-linear. Metabolism, how kinetics is happening, whether any carcinogenicity or mutagenicity is happening or not, because we all know these days the example of what we call the Johnson's baby powder, the company was sued by a few citizens in the US because it was causing carcinogenicity when it was applied to babies. So that comes only when we have sufficient data on babies available. So that has not been considered during preclinical studies. So similarly, in phase 1 clinical trial, we do it on very limited population below 100. Healthy volunteers are being tested on this. Similarly, phase 2, this is generally done on 150 to 350. These days, people prefer doing phase 2 clinical trials on more than 1,000 patients. Afterwards, we do phase 3. In phase 3, we generally prefer having it more than 4,000 these days to get more varied patient groups to determine short-term efficacy and safety in the formulation. So once phase 3 is done, then we do the registration. Now, when the registration is done earlier, we never used to have phase 4. Phase 4 is like post-approval of the medicine, how the patients are taking that medicine, whether any side effects are happening or not, whether any clinical issues are happening or not. So those were not there earlier. But because of thalidomide case, the after registration, the phase 4 post-marketing or post-approval what we call studies are also being done. So this when this happens, it also helps in spontaneous reporting. So that is being done through pharmacovigilance. So pharmacovigilance is an integral part of drug development process, right from phase 1 till the post-marketing. So it is a continuous process. We cannot stop this pharmacovigilance process when the medicine is there in the market. So why it is needed? I've already explained that there are rare ADRs like 1 in thousands or 1 in 1 lakh, 1 in 10,000, which are unlikely to be identified during pre-marketing studies. So this can be toxicity to reproduction, like impotency can happen, genotoxicity can happen, carcinogenicity can happen. So therefore, pharmacovigilance is very much necessary when we are studying these issues. A drug that belongs to a widely used pharmacological class may be used in up to 1 lakh individuals within the first month, so that a rare but serious ADR may occur only in few patients. So but that is not possible when we are doing phase 1, phase 2, phase 3 clinical trials. That generally happens only when we market the product in the market. What are medications error? Medication error generally are considered when these are being done at patient's end. So it is any preventable event that may cause or lead to inappropriate use of or patient harm while the medication is control of the healthcare professional, patient, or consumer. Like I have to take one tablet, but because the pain is so much, I'll tell, okay, no, let me take it. Let me take 2 tablets so that my pain is getting relieved within 1 hour instead of 2 hours. But that generally doesn't happen. Patient doesn't know whether there is going to be any side effect of the same or not. Like this is especially serious when the patient is having trauma, when the patient is having anxiety, depression. So when the patient is having depression, he or she might take multiple tablets. So that is also going to cause death also. So these are nothing but medication errors which we all right now know that sometimes are getting committed even from a normal human being, because we are unaware what exactly the issue will be if we start taking medicines in a wrong way. So what is key fact? No medicine is risk-free. Even when we are taking the safest medicine like paracetamol, it can have side effects like GI upset in few of the individuals. It can cause acidity in few of the individuals. So vigilant assessment of the risk and benefits of the medicines promote patient safety. People in every country of the world are affected by ADRs. It doesn't mean that India people are strong, so the adverse drug reaction might not happen. It doesn't happen in that way. Adverse drug reactions can be of anything. It can be GI upset. It can be stroke also. It can be chest infection. Anything can happen. So what is it related to? It can increase the cost. It can have hospitalization. Surgery can also be done, and also loss of productivity. We all know these days people are suing medical companies for medicines which cause loss of what you call productivity, in the form that people don't memorize the things now as they used to do before the taking medicines. So these are the issues which we normally face. So these can also cause deaths. So we can say whenever we are taking medicines, we have to be very vigilant how much medicines needs to be taken, and that should be taken only when it is being prescribed by a registered medical practitioner or by the doctor. What are medications error? In USA, drug-related death rate is the highest which we see, because once a product is being developed, first it is being developed in the US, because the cost of developing a medicine is being recovered maximally through US, and that is why we see highest adverse events in US only, like 19%, out of which what the data shows is 50% is preventable. So you just assume if we prevent this 50%, how much cost of treating this adverse events can bring down, can be brought down in a patient. Similarly, in UK, because after US, UK and Australia are the second largest markets, adverse drug events are contributing near about 10% of the overall patients which are being treated. Again, 50% is preventable, but you can just assume 1,100 deaths are occurring in UK just because of the adverse drug events. Similarly, Australia, it is nothing but 16.6. And according to the data, it is said that 2 to 4% are the people which are having adverse events are being admitted in hospitals also. So out of which near 75%, which is way higher than US and UK. So 75% are having adverse events which can be prevented. So the key facts are the majority of adverse events are preventable and can be due to wrong diagnosis. It can be due to prescription, undetected medical or genetic allergic conditions, self-medication. In India, generally all are doctors. All people are there, they take their medicines or their own. So self-medication also is an issue in India. Not following instructions for taking the medicine. If the doctor says you have to take half a pill, then people take full pill just to avoid these issues, which doctor has already prescribed. Reaction with other drugs, they generally don't. In India, generally people generally don't tell what exactly are the reactions which occurs when they take the medicines. So that is also going to cause the adverse drug reactions, like people who are allergic to lactose. So they should avoid taking medicines like rosuvastatin, which are having lactose in it. So that will cause nothing but allergy to happen. Similarly, use of substandard medication. We generally go to the chemist and will say, okay, do we have any alternatives for this medicine which is having high cost? So he will give a medication maybe from a company which uses a substandard medication. So these are all the adverse drug reactions which can be prevented, but it occurs in every individual. So it can be categorized into three different categories. 60% are because of prescribing and transcription. Administration causes near about 30%, and dispensing causes near about 10% of adverse drug reactions. A&M responsibilities, we have to early detect the unknown safety problems, detection of increases in frequency, identification of risk factors, quantifying the risk, reporting, labeling, and approach. Of course, when we all have a medication being prescribed, we generally also get the package along with the medication. We get the package inserts. We all should go through about it. We should see how much medication needs to be taken, what are the medication we should avoid taking when we are taking that medication, what all things needs to be taken along with the medication, whether it is to be taken with milk, whether it is to be taken with the juice, whether it is to be taken with the applesauce. We all should follow the pack insert. So when does PV start? Testing of drug safety begins always when the clinical trial happens. So I've already explained this to you. Right from the start of clinical trials till the medicine is there in the market, the pharmacovigilance should be there. Without that, the issues of adverse reactions can happen. So adverse drug reactions, possible causes. So what causes adverse drug reactions to happen? Intrinsic factors like pharmacological effects, idiosyncrasy. Idiosyncrasy is nothing but like if I'm taking cetirizine, I might be totally conscious, but some people doze off. They say that I get sleep after taking cetirizine. So that is nothing but idiosyncratic reaction. Similarly, some people may get carcinogenicity or teratogenicity. These are intrinsic factors of a drug. So that is why some guidelines have come up from US FDA, especially ICF M7, which talks especially about the genotoxicity of a molecule. Similarly, extrinsic factor, adulterants, contaminants, like we are taking spurious drugs, spurious excipients to manufacture their drugs. Underlying medical conditions. When we take a medicine, we never tell to the doctor, like I am having heart issues, I'm having diabetes, I'm having kidney, give the medicines. But we generally don't reveal this to the doctor. Interactions, whether I'm allergic to any food, allergic to any medicines, those should be also a possible causes for adverse drug reaction to happen. So classification I'll give. Pharmacovigilance, it can be active, it can be passive. ADS can be because of cause or it can be because of severity. So what is passive or active pharmacovigilance? Active pharmacovigilance is active safety surveillance, means that active measures are taken to detect adverse reactions. So we upfront take active follow-up with the patient. So that is generally being done when the patient is under clinical monitoring. We in clinical trials, in hospitalization, So if any adverse drug reaction happens to a patient during hospitalization or during a clinical trial, the doctor is there to support the patient, and accordingly they can tell what are the symptoms in the patient, and accordingly active pharmacovigilance can step in. So what is passive pharmacovigilance? Passive pharmacovigilance is nothing but the companies which are manufacturing the drugs, they touch base with the doctors. These doctors report that the patient had come yesterday and he had severe diabetes, and because of that he had higher blood sugar count when he has taken dexamethasone, because we all know dexamethasone causes the sugar levels to increase in diabetic patients. So these are nothing but passive pharmacovigilance approaches which we generally do when we talk about pharmacovigilance. Adverse drug reactions are categorized into 6 different categories. The most hyped one or most talked about is the type A, which is because of intolerance or side effects. Bizarre effects are rarely spoken about, which is because of idiosyncrasy. Type C can be chronic effects, type D delayed, type E end of treatment effects, and type F failure of therapy. So type A, when we talk, mostly we talk about type A and type B only, because type A and type B generally talks about intolerance and side effects and bizarre effects. So type A is intolerance and side effects which we generally see, like cough with ACE inhibitors. When we take ACE inhibitors like enalapril, captopril, perindopril, so when we take these kind of medicines, cough is a common phenomena. So these are nothing but adverse reactions. Similarly, type B can be because of some serious side effects which can be life-threatening also. So when is an ADR serious? ADR can be serious only if it harms the patient, like death, life-threatening. So hospitalization, you'll say, okay, nothing, hospitalization is only happening, nothing wrong is happening in that. But you just see the trauma the patient faces when the person goes to the hospital, the cost which is getting involved in hospitalization. Accordingly, some people may have disability. Like we have already seen the case of thalidomide, persons or the babies which were born were having deformed limbs. So that is a disability. Congenital anomaly, required intervention to prevent permanent impairment or damage. Like some people can have ototoxicity or they can have permanent ear functioning also. Permanently damaged ear functioning also. Similarly, malignancy and malformations. So all these are categorized in serious adverse drug reactions. Serious adverse drug effects are nothing but the ones which can cause deaths also. But when we take serious adverse events, why it is not considered as severe adverse event in a study when a death occurs in a stroke, when a medicine is used to treat a stroke trial. Like I'm having a medicine which is going to get launched in the market. I want to run a clinical trial. So that will be generally done on patients which are having stroke issues. So if I'm dosing that medicine to a patient having stroke issues, and if he dies because of stroke, I'll not say that it is a adverse event. It can be because the medicine has not worked also. Similarly, in cancer trial, when we are developing a injection like paclitaxel injection, if I'm giving it to a cancer patient and if it doesn't work and the person dies because of cancer, so I'll not say that it is a serious adverse event. I'll say that maybe the medicine has not worked. So exceptions to hospitalization as severe adverse events. In case or absence of any medical adverse events, administrative admission, social admission, so that can not be related to a severe adverse events. That the event which is not related to the medicine is called as exceptions to hospitalization as severe adverse events. Confirmation of case seriousness. Seriousness should be changed if the case received a serious from a healthcare professional, received from a licensed partner or regulatory authority, clinical trial case that already reported as serious. MEDRA, we'll talk about MEDRA in the next presentation. Preferred critical term, coded as always serious. So only the cases which are serious should be reported generally by healthcare professional, licensed partners, or the companies, or during clinical trials, or through MEDRA. These are the only 4 ways how we can report a serious adverse event. So ratings by IGARD. IGARD is a non-official scaling which exists to communicate overall drug risk. It generally has categorized the seriousness into different categories. High risk, which is red, like adriamycin and with vincristine, these are being used for anti-cancer patients. And but these are having some adverse events, like adriamycin can cause heart-related issues, but because its benefit is quite high as compared to heart issues, so this is being prescribed like a patient who is dying because of cancer, even if that person is having slight heart issues because of the medication which is being given, so that can be still considered as benefit, but it poses a high risk. Similarly, we categorize it as orange only when a personal risk reduction plan is there to be taken care with the doctor, like prednisolone, warfarin, insulin. We know that when we are taking insulin, like our grandparents or our mothers, fathers, when they are taking insulin, those patients can have high or low blood glucose levels, but those cannot be considered as high risk. That can be considered as elevated or orange risk because we know how to control it. Similarly, some drugs are being categorized as yellow, like Januvia. Januvia, we all know that these are nothing but anti-diabetic medicines, so they can lower the blood sugar level, but that can get lowered to a very low level. But we know, okay, we take sugar candy, or if we take sugar, that will again come back to a normal stage. Similarly, general lattice blue, like Lipitor. We all know Lipitor, that is atorvastatin. That can be taken by a normal patient also. If it is being prescribed by the healthcare professional, we all know even if any side effects are happening, that is not a serious side effect because the safety and efficacy of that medicine is well established. And of course, the green one, we are all taking vitamin C because of the COVID issues. We know that it is having no harm, and that is why it is being categorized as green category. So we have all talked about adverse events, adverse drug reaction, unexpected adverse drug reaction. Similarly, suspected, unexpected, serious adverse reactions. So these are nothing but the definitions of the same. So common examples I'll give, like amidopyrin. When we give, it causes white blood cell disorder. Cleoquinol, it causes visual impairment. Oral contraceptive, it causes thromboembolism. Statins, it causes muscle degeneration. Statins, especially simvastatin. Now it is being used very rarely. Thalidomide, it can cause phocomelia. So these are common adverse drug reactions which everyone knows. Active ingredients which are withdrawn from the market. Since the adverse drug reactions are there in the patients, those which can be taken care can still be considered as manageable adverse drug reaction. But those which cannot be managed and which can cause severe side effects like deformity, carcinogenicity, heart issues, stroke. So those medicine cannot be continued. So examples of these are like thalidomide, benoxaprofen, fenformin, which was we all know about metformin. Metformin is being used to treat diabetes, but there was one medicine called fenformin also, which came afterwards, but it caused severe adverse drug reactions, so it was removed from the market. Similarly, rofecoxib, valdecoxib, these were also removed from the market because of serious side effects. So, pharmacovigilance reporting. Pharmacovigilance reporting requirements are identifying the minimum criteria for AEM, adverse event monitoring, reporting, and identifying the HCP reports. So these are the main pharmacovigilance reporting requirements which are being practiced overall by the healthcare professional or by the industries. So minimum criteria for AEM reporting. When we say AEM reporting, that is adverse event monitoring. How do we identify AEM? It should be an identified patient, like a person. When it goes to the doctor will identify, okay, Mr. X had come to me last month, and he was taking, say, X product, and because of that, adverse reaction happened, and that is why the reporting was done. So this is a minimum requirement, like patient, who was that patient, if possible, what was the age, what was the medicine which that patient had taken, what was the adverse event, and how it was identified. So components of a case report: identify patient. So at least one of the details should be available, like age, date of birth, sex, history, case identification. Product clearly refers to name of the drug or biologics or any device. If I'm reporting a pharmacovigilance adverse event and I don't know what the product is, so it doesn't mean anything. If I don't know who the patient is, whether it's a male or a female, what age group he or she was, that is of no issue. That is of no use. Similarly, adverse event, whether it was diarrhea, whether it was heart problem, so that is again of no use. So these are the 4 structural pillars which are required when we are doing a case reporting in pharmacovigilance. So these are the entire contents of ICSRs, which we'll touch base upon in the next presentation. Who can report? Now you all say that I cannot report what you call adverse events. So whom should I consult? You can consult it to the physicians. You can consult it with the dentists. You can consult with the nurses when you are hospitalized. You can also consult with the pharmacist, so that when you go to the pharmacist and you tell them that I'm having this reaction because of this medicine, they can report that directly to the company. In India, generally, it doesn't happen in that way, but now after 2004, in India also a proper pharmacovigilance reporting has started, and it is still in nascent stage, not in the advanced stage as happening in US, Europe, or Australia and New Zealand, but still it is being developed, and people are reporting these issues in India also. How to report? Like, you can report adverse reaction by formal communication, by going to the healthcare professional personally, or you can use a telephone. When you are taking any medicine, you know which company it is. You can go to the company's website. You can have the company's number. You can call them and report it. Similarly, you can do an email. Along with that, you can give all the attachments, like what all issues you are facing. You can attach the reports, and that can be directly mailed to the pharmacological centers of that company, or it can be done through mail communications to pharmacovigilance systems set up in India also. Similarly, fax can be done, internet, anything. So you have to just report. Reporting can be through anything. You can do WhatsApp, you can do text messages. It is just a form of reporting only. But that should all contain these basic 4 pillars. Of which to report, like medicine products covered by pharmacovigilance activities includes centrally authorized through mutual recognition procedure, purely national authorized, new medicinal products, old medicinal products, biologicals, vaccines. So anything, any medicines which you are taking, that shall be reported. Be it vaccines, be it injections, be it nasal products, be it balms which you are using, anything can be reported. So what to report? That when I'm going to report, I should tell, okay, this is not affecting. Okay, I'm having a resistance to it. It is having an interaction. I'm getting dependent on this medicine, and I'm having unexpected beneficial effects. So these can be reported very easily. And what can be excluded? Now, excluded doesn't mean that it is to be excluded every single point of time, but this should be excluded only if it happens because of the patients, like therapeutic failures. Patient is not taking the medicine. If it is not taking the medicine, then the effect will never be there. So these are therapeutic failures. Similarly, unintentional or accidental poisoning, abuse. So if you are taking overdose and because of that some adverse event is happening, you cannot say that it is because of the medicine, though it can be reported, because we then will come to know, okay, if it is taken in higher concentration, it can cause accidental poisoning or abuse, but it will not be considered as severe adverse event. Similarly, in errors in administration, if I have to take only one single tablet and I'm taking 5 tablets, so that is error of administration. Similarly, non-compliance. The doctor has prescribed you to take a tablet twice in a day, like after lunch and after breakfast. So you are not adhering to that. You are taking after lunch and after dinner. So that is a non-compliance which you are doing. So for that, we'll exclude it, but you have to report it so that the data can be gathered. For new drugs, report all suspected reactions, including minor ones. When to report? A report should be completed as soon as possible. Normally, it is said that within 24 hours, when the event has happened, it should be reported to the authorities so that the action can be taken immediately. And in some cases, the entire report has to be within 7 days, because the event can lead to death also. So it is better to advise reporters not to wait until final results and information such as hospital letters are received. These additional details can be sent to the pharmacological center later as a follow-up item. Follow-up when necessary. This is very much important. When a severe event is happening and it is not complete, then we should always follow up, because then only we'll be able to know whether that was a short-term adverse event, or it was a serious event, or it was a non-serious event. So examples can be a request for essential missing details, information on the final outcome, like we'll call to the patient and we'll ask whether any adverse event is still prevailing in that patient or not. Similarly, a re-challenge. What is a re-challenge? We'll discuss this in the upcoming presentation. The results of laboratory test, whether a person having diabetes was having severe issues. So if he or she has carried out a medical test to determine the blood glucose level, so that can be also taken care in this. Where to report? I've already told that it can be done through the pharmacological centers, it can be done through medical professionals, it can be done through clinics, hospitals. They should be stored securely to maintain confidentiality. We shouldn't be circulating it amongst our friends. It is only limited circulation which is available for these documents. Who are the partners? The partners are government, of course, industry. Industry means the pharmaceutical industry or the hospital industry, hospitals, academia, medical and pharmaceutical association like IPCA, IPAC, IPA, poison information centers. We all have poison information centers situated in multiple cities, so we can tell them also. Healthcare professionals, patients, consumers, media, and WHO. So how do we go about PVG reporting. So once report is being processed, the case is being received at the company's end, inventory is being made, and then the distribution happens. So if the company is in US, so it will be sent through international divisions to various locations. It can be US, it can be UK, it can be India, anyone. Then a triage is being found, and then medical coding happens. So when the medical coding happens, the quality assurance people will code that information, and the information will be stored in a system. Then it will be also reviewed by a medical reviewer, and when the information systems are being compiled, that will go directly to the research and development, where the safety and efficacy of the medicines will be challenged, the adverse events will be challenged, and then it will be reported to health authorities, because ultimately the health authorities are the one who will call all the shots. They will determine whether the medicines are effective or not, or whether we should withdraw the product from the market. And similarly, they also publish the PSUR publications. The functions of pharmacovigilance. As per WHO guideline, the pharmacovigilance should detect and study the adverse events, adverse drug reactions. Measurements of risk and benefit should be identified. Similarly, measures of effectiveness and monitoring the impact of any corrective actions should be taken. Benefit and harm evaluation needs to be done. WHO Program for International Drug Monitoring. It has a separate policy which governs the pharmacovigilance program. It exchanges the information. It sends the technical supports to various countries, especially the developing nations like Africa, Asia, and even some Middle East countries. They set up they have an advisory committee on safety of medicinal products, and accordingly they'll decide what to do in case of an adverse event which is happening in a patient. Exchange of information from WHO generally happens through newsletters, drug alerts, drug informations, restricted pharmaceutical lists, and there is a Vigiment electronic exchange which is happening. There is a site, and similarly a signal can be produced because of this. Technical support to the countries. I've already told that WHO provides technical guidelines on all aspects of pharmacovigilance, especially to the underprivileged countries like Africa, some South Asian countries, and Egypt also. It also provides training courses on pharmacovigilance, which a person can go through, and accordingly they can apply for as a pharmacovigilance healthcare professional. So why WHO is necessary? It has a Uppsala Monitoring Center where all the collaboration or where all the compilation of the data of adverse drug reaction is being compiled. Each year it is said that more than 3 million reports are being generated, and right now I'll say that near about 250,000 cases are increasing per year. So you can just imagine what we are talking about in 2025 will be near about 5 times what I'm right now having. So if I'm having 1 lakh cases a year increase right now, maybe after 5 years it will increase to 5 lakh cases in a year. So that is why you just imagine how developing this field of pharmacovigilance is developing, and that is why even in India this is being taken care very seriously. However, still it is in a nascent stage. The countries like US, UK, Germany, Australia, and Canada are the top 5 reporting countries. So these cases being reported by these countries are right now being taken care. But that doesn't mean that in India or our neighboring countries like Pakistan, Bhutan, Nepal are not providing the reporting structures in their countries. Those are very much there, but still in a nascent stage. So ADI reports generally have analysis being done, data mining is done, output is being sent to national centers. They are producing ad hoc research results also in the form of publications. WHO right now is having publications for various vaccines also, which are being produced for COVID. Similarly, signal documents are also being produced as a adverse reaction reporting structure. So future challenges, especially when we say about the developing nations like India, Africa, awareness is still not there. Like in metropolitan cities in India, the awareness is still there. People are right now aware through social medias or through what we call WhatsApps, LinkedIn's, Facebooks. They are very much aware what is happening right now, and that is why they are very much aware. But still, people who are in villages on in tribal areas, the awareness is still not there. So these are the challenges which WHO or even the healthcare professional are being facing daily. Similarly, monitoring all the medicines is still a biggest challenge which we are facing. Integrate work through WHO is very much important because World Health Organization is the one which coordinates with all the neighboring countries, with all the developing countries, with countries which are totally developed, and from that developed countries, the information is being passed to neighboring countries or to the underdeveloped countries, so that chain of network regarding the adverse drug reactions can be established. So at the end, I'd like to thank you with a note by Shakyamuni Buddha, which says, "People who are vigilant do not die. People who are negligent are as if as they are dead." So we have to be very vigilant because that will take care of all these adverse drug reactions which are happening with the medicines. But if we are negligent, it may cause death also. It can be of our friends, it can be of our relatives, it can be of anyone who are our nearer and dearer ones. So thank you for the presentation, and any questions are there, I can take up after the next presentation, which is being delivered. Thank you.
Hello, am I audible? Ramandeep sir? If the participants are having any questions, they can place over here in the chat box. Okay, I think no more question. Okay. Thank you for this session, sir. So hereafter, we are moving for the second session, which will going to be delivered by Dr. Manpreet Kaur Madam. So our second guest, Dr. Manpreet Kaur Madam, is having near about 9 years of experience in pharmacovigilance and drug safety, including triage, data entry, data validation, and QC. Currently, she is working as a senior drug safety specialist at Excel India. She has also worked with Quantum Solutions as pharmacovigilance specialist and pharmacovigilance scientist. Dr. Kaur also trained various pharmacovigilance scientists as well. She is having wide experience in various areas such as oncology, infections and infestations, dermatology, and many such areas. Basically, she is a dentist as her core profession. So, with this brief introduction of our second speaker, I would request Dr. Kaur ma'am to take over the session further.
Thank you, sir. Thank you for your kind words. Is my screen visible?
1 minute, ma'am. Let me make it visible.
Yeah.
Yeah, it is visible now.
Okay. So the introduction to pharmacovigilance was very well explained by Dr. Ramandeep. And now I'll further explain the methodologies in pharmacovigilance. So first of all, I'll start with the methods which are used in pharmacovigilance. The first is the spontaneous reporting, and this is the very simple reporting which can be done by anyone, by the healthcare professionals, by the consumers, by the patients, relatives, anybody. It can be in the form of legal reports which are reported via lawyers. Then further reporting is in the form of clinical studies data. The clinical studies are carried out, the clinical trials on the drugs which are still in the developing phase. And then further the solicited reports are there, which are via the patient support programs, which help the patients via money, and they participate in those patient support programs and provide the data. Then literature reports are there, which we get from the literature data, and the registry reports, which are basically the post marketing phase of the drugs. Then the case control surveillance is there, and then is the automated population databases, or you can say data mining. These are the automated population databases in which anyone can record the data, and we can extract the data from there. Then people use a lot of medical devices. The medical devices also have complaints. So this is another method in pharmacovigilance. So spontaneous reporting, though it has advantages, but it has limitations also. The advantages are that it has a wide coverage, like all patients, all drugs, and all adverse drug reactions are covered up because it is reported by anyone. And it is effective, it is continuous, it's rapid and cheap also. But limitations are also there because they can be underreporting, and no quantitative assessment is there. Sometimes there is no proof of causality also, and the comparison of drugs is also difficult. So further testing and evaluation is required in spontaneous reporting. The most important part I'll discuss here is product labeling or the listedness assessment. This is basically the products which we are taking. They have their warnings, precautions, their indication, and everything is recorded in their labeling documents. And we are using two types of documents, which is United States Package Insert, that is USPI, and the core data sheets. It has all the summary of product characteristics of that particular medicine. So this is the basis of pharmacovigilance by which we judge if the adverse event occurring is already listed in their labeling documents or it is not listed. How the drug is working over the years. So there are two types of adverse events. We consider it as expected or unexpected. Expected are those which are already listed or contained in the labeling documents, and the unexpected are those which are not there mentioned in the labeling documents, which develop over the years with the use of the drug. So listedness assessment, How we assess the listedness, it is done by the dedicated medical professionals, by the physicians, and they check all the parts of the labeling document, all the undesirable effects mentioned in the document, warnings, precautions, the contraindications, their effect on pregnancy and lactating mothers, the overdose, everything is checked, everything is reviewed, and then the product is, the adverse drug reaction is labeled as listed or unlisted. Then further comes the causality assessment. Causality is the relationship of the suspect drug, that is the medicine we are taking, with that of the adverse drug reaction. So this is a very, very important part. We have to assess it as related or unrelated. And it also tells you how close is the relationship of the drug with the event, and did our medicine or our drug actually has caused the event. So if the adverse event is like it does not have any temporal relationship with the product being taken, and we know there are some underlying causes also, which the underlying diseases which the patient is experiencing. So if it is clearly not related to the medicine, it comes up as not related. Unlikely related is when we assess it as unlikely related when it is doubtfully related to our medicine, like some lab abnormalities are there, and some other underlying conditions we can see which make it as unlikely related. And then the possibly related, possible in this, it has a reasonable relationship, the medicine and the adverse drug reaction, they have a reasonable relationship, but the adverse drug reaction can be due to another or equally likely cause. So we say it as possibly related. And in probably related, it increases the temporal relationship as compared to the possibly related. And we say it as definitely related when it has a direct relationship and there is no other cause which explains that adverse event. So we call it as definitely related. Then we have medically confirmed reports, the reports which we are receiving from the healthcare professionals, and they confirm the occurrence of that adverse event, and also the causality is definitely related. So those reports are called the medically confirmed reports. Further, I'll discuss the special case scenarios. Not only adverse drug reactions are, see, we are having some stomach upset or we are having headache or stroke. These are not only the things which are reported. There are some special case scenarios also. These are not directly related to the adverse event, but they involve a circumstance that increases the risk of the patient and risk of the patient that they may further develop the adverse events in due course of time, like the drug misusage is there, some extravasations are there, medication errors are there, overdose is there. Sometimes the patient is taking insulin injection and the drug and all the medicine spills over, so the patient is not able to take the complete medicine. How will it affect the patient? It leads to hyperglycemia. So that is this is a special case scenario which may lead to adverse drug reactions. And sometimes the patient is misusing the drug. The patient is taking the drug twice a day, rather it should be taken once a day. So that is the misuse of the drug. And overdoses, the patient is getting benefited from that drug. The patient is taking that drug more frequently. So that leads to the overdosage. Then drug interactions, it has already been discussed in the previous presentation also. The drug interactions could be there. Sometimes the patient is taking two medicines together and it leads to the adverse events. Then comes the lack of efficacy. Sometimes the person is taking the drug, but it has no effect on that drug, on that underlying disease like the hypertension or the diabetes. So that is considered as the lack of drug effect. That means the patient is not having any benefit from taking that drug. And sometimes the patient is taking the drug as in unapproved schedule, like the patient was asked to take the drug in the morning, but the patient is taking the drug at night or is missing doses. So these all are considered as the special case scenarios and must be reported because they lead to the adverse events in over some time, in due course of time. Then I'll discuss about the narrative writing. This is a very important part in pharmacovigilance reporting. So what we do here is we make a written summary of all the clinical information and all the relevant information relating to that medicine and the adverse event. So what all key elements are there in the narrative writing? First we have to mention the source type from where the data we have collected, and then at what time the data is collected and at what time the data is followed up. All this have to be mentioned in the top. Then we have to mention the complete patient details, complete like the patient's demographics, and then we mention about the patient's medical history, all the past drugs which the patient is taking. Then we mention in detail about the suspect drug. Suspect drug is the drug which is in question, like from which the adverse events are occurring. And then we have to also mention about the other concomitant products which the patient is taking for the comorbidities or the other problems. Then we mention about the adverse event in complete detail, that when it occurred, like we have to mention the start date, then we have to mention if we have taken any treatment for that, then we have to mention what is the outcome. And further, we also do some laboratory test to prove the diagnosis or all that. So we have to completely mention that also. So this gives you a total summary of the adverse drug reaction and the drug of the complete case. And this should always be in a chronological order and should always comply with the standard operating procedures. Then I'll discuss in detail about MEDRA. This is very important. It is the medical dictionary. It is the medical dictionary for the regulatory updates. And it provides a classification for the wide range of clinical information, able to support for multiple medical product areas. And this is actually developed so that all the people all over the world are on the same page. It is it should be internationally same everywhere. And to standardize the communication between the pharma industry and all the regulatory authorities. And it can be used through all the phases of the development cycle. So, so what is there in MEDRA? All the diseases, all the diagnoses, signs and symptoms, we can link them through MEDRA. All the therapeutic indication of the suspect drugs and all the lab investigations, their names, the medical and surgical procedures, and when we code the medical history of the patient, all we go through MEDRA. For example, I'll show you. If the patient is like headache, it is of various kinds. The etiology can be different for the headache. So sometimes headache can be due to the lumbar puncture, right? So we we search for the headache in the MEDRA dictionary. We get all the results, and we have to just click on the result which is related to the our case scenario. So in this case, the headache is due to lumbar puncture. So the low level term comes as headache lumbar puncture, as you can see, and it falls under the nervous system disorders. There is a preferred term, there is a high level term, there is a high level group term. So we get to know that it falls under this type of system organ class. These are the various system organ classes which are covered in MEDRA. They're basically 26. All the cardiac disorders, all the blood related eye disorders, GI disorders. So everything is included in this, be it pregnancy conditions, psychiatric disorders, musculoskeletal. So all are included in the MEDRA. This is again a basic structure example of the MEDRA linking. The LLT term is there first, multiple personality disorder, then PT is dissociative identity disorder, further to the high level term, and to the more wider HLGT, dissociative disorder, and SOC is the most widest term, that is the psychiatric disorder. And what are the implementation benefits of MEDRA? It saves our valuable time, it saves money, it saves resources, and it decreases the guesswork also, because we have to link to the most nearest linking term, and so it decreases the guesswork and it leads to standardization of the data set, which is being followed all over the world. And MEDRA is updated biannually, once in March and once in September. Data assessment in pharmacovigilance is through the individual case report assessment, which we call as ICSR, aggregated assessment, signal detection, interactions and risk factors, serial like clinical studies, and frequency estimation. I'll discuss in detail about the signal detection, which is the most important thing, which is the basis of pharmacovigilance. Whenever we receive an alert from any available source that a drug may be associated with previously unrecognized hazard, like some of the adverse drug reactions are already contained in their labeling documents, or that a known hazard may be quantitatively, that is, it is more frequently occurring, or qualitatively, like it is more serious, it is more severe, and it is different from the existing expectations. So that detects a signal. The minimum requirement is a single well-documented report with a positive re-challenge or replication of findings in a series of reports. And what is the SNP rule for the signals? It should be strong, it should be new, important, and preventable as well. A signal consists of a hypothesis and data confirming that hypothesis, and then arguments in favor or against. Data of the signal should be qualitative, that it should be clinical. We must know what is the severity of that adverse drug reaction which we are reporting, the signal which we are reporting. It should be quantitative, epidemiological, how many times it is occurring, in how many people it is occurring, everything should be there, experimental, and it is developing over time. When is a signal likely to be relevant? If it is an early warning, it's a new adverse reaction with a new drug, if it has to be seen via the public health perspective, if it is a very important drug, like some drugs are being taken by a mass number of people, like for a serious indication, which is very, very common for a stroke or for diabetes, any such drug, which is widely used. And if it is causing some very, very serious reaction, and it is in a large number of cases, and it is alarming when there is some rapid increase in the reporting, then regulatory intervention or prevention should be there. There is a benefit risk assessment, and if there are changes in the benefit risk assessment, that leads to a signal. Scientific or educational value should also be considered. Then signal follow-up should always be there, like for the drug exposure. If the development over time of the quantitative data, like if the data is increasing over time, both quantitatively and qualitatively, then signal must be followed up. Reporting distribution should be there, best case, worst case scenario, and targeted comparisons should be there. And then case control studies, the nested case control studies should also be followed up. Now, when we receive a signal, then it should be acted upon. The internal communications are done via the national centers of the countries. UMC is the Sauna Monitoring Center, which is the WHO collaborating center. It is in Sweden. Then via the pharmaceutical companies and academia, that is where extensive research is taken care of. Then signal testing, further evaluation is started of that study. Then further, the regulatory action will be taken, That is the labeling documents would be changed, data sheet changes will be there, and further the last action would be external communication. External communication is through the drug information centers, the national bulletin, and the publications. This is the picture of a signal document. This is sent to all centers for the national distribution, individualized section available to industry, and all recipients are encouraged to comment on the topics presented. Statistical analysis allows the pattern recognition in large amounts of data, also incomplete case data, and allows identification of interactions and syndromes. This is the risk-benefit analysis. So we all know that drugs have benefits. All the medicines which we are taking should have benefits, but risks are also there. And risk should never outweigh the benefit. Risk-benefit analysis is the comparison of the risk of a situation to its related benefits. For research that involves more than minimal risk of harm to the subjects, like to the people, the investigator must assure that amount of benefit clearly outweighs the amount of risk. Only if there is a favorable risk-benefit ratio, a study is considered ethical. The risk can be anything. It can be physical, it can be death, it can be disability. It can be psychological also, depression and anxiety. It can be social, like discrimination, and it can be economical also, effects like the jobless, the people get jobless because of some deformities or something like that. So the benefit risk evaluation is when it is done, what is the action taken? We edit the product information like indications, usage, the dosing instructions, the contraindication interactions, warning and precautions. Everything is edited. And if the risk is very, very high as compared to the benefit, we withdraw the marketing authorization. It is withdrawn for that particular product. Risk analysis. We have to identify the risk in the preclinical studies, harms identified in clinical trials, and formal mortality and morbidity study. Then we have to understand the risk. The rigorous case definition, case series analysis, and clear description is in the label. We have to understand the risk completely. Further monitoring of the risk is required. That we can do in the post-marketing studies, in the post-marketing surveillance, when the drug comes in the market, and through via various cohort studies and the registry studies. Risk mitigation. We have further, if we have collected the data, then we have to communicate it through, how can we do that? Through the advice in the label, via the partnership with the regulators, regulatory authorities, and through the education of the physicians, the patients, the company staff, everyone. And the act to reduce the risk, limited distribution, limited prescribing rights, contraindicate for certain groups, indications or routes of administration, and advice for the high risk group. This is how we can act to reduce the risk. Then further, we have to measure the outcome of the interventions taken, like the corrective therapies which have been taken. We have to measure the outcome. Now, this is the post-approval risk management plan. What is its objective? Its objective is to quantify the risk, and then it requires the understanding of the numbers needed to assess the risk, particularly the rare risks. And it is important to consider how to identify the immunogenicity risk. Framework should be proposed for ensuring attribution of detected risk to specific product. Then I'll talk about the regulatory bodies. You must have heard in the earlier presentation also about the Uppsala Monitoring Center. It is actually the WHO Collaborating Center for International Drug Monitoring. It is in Sweden, and it takes care of all the drug safety internationally. Then there is EMEA. This is basically a European regulatory body, which includes the Europe, Middle East, Africa, Middle East and Africa. These countries are involved in this. So what it do? It is involved in setting the standards, like guidelines for the pharma industry, so that everybody, all the pharma companies are on the same page. It supports the companies during research and development. It provides the scientific advice to them. Then it works for the medicines for rare diseases, and it supports the companies prior to the applications. It further, in my opinion, how it is implemented through the European-wide countries. And then the safety monitoring is done via the post-marketing applications and surveillance. Inspections are done and information is provided to the patients and healthcare professionals. Various pharmacovigilance regulations are there. Medicines for Human Use Clinical Trials Regulations 2004. Legal requirements. This was developed in 2004. EU directive and detailed guidance required harmonization of laws, and there is some flexibility also as per the changes it is updated. Then there are some internationally accepted principles of good clinical trial practice reporting, E3, E6, E8 and E2A. The further E2 clinical safety, it has the E2A, it has definition and standards for expedited reporting. E2B, data elements for transmission of adverse drug reaction reports. E2C relates to the PSUR, periodic safety update reports. E2E is pharmacovigilance planning. And this all is taken care of or helped by the MEDRA dictionary. M1 leads to medical terminology. M2 is electronic standards for the transfer of regulatory information and data. And M5 is data elements and standards for drug dictionaries. The pharmacovigilance specification is actually the summary of important identified risks, important potential risks, and important missing information about a product. Now, pharmacovigilance in India. India has become a major hub for the clinical trials. It has extensive pharma industry, but in pharmacovigilance, it is still lagging behind. The developed countries, the 5 countries which were discussed in the earlier presentation, they are leading in pharmacovigilance, but India has also started doing a lot in PV, but still it is lagging behind. India's Drug Control Department within the Ministry of Health and Family Welfare initiated the establishment of nationwide network in 2004. So different PV regional centers have been listed in this below site. So where to report? It has already been discussed. Local area reporting centers, anybody can report there. Then it goes to the peripheral PV centers, further to the zonal, and then to the regional PV centers for the causality assessments and all. And then Central Drug Standardization Control Organization further gives all the data which is managed by the Uppsala Monitoring Center in Sweden, that is the WHO Collaborating Center. Now, pharmacoepidemiology, the study of the use and effects of drugs in large groups of people. Epidemiology can be defined as the study of the distribution and determinants of diseases in population. These studies can be divided into different types, which are descriptive studies, describes the disease or exposure, and may consist of calculating the rates, like the incidence and prevalence. Now, there are good pharmacovigilance practice and effective pharmacovigilance. Good pharmacovigilance practice is based on acquiring the complete data from the spontaneous adverse event reports. And these reports are used to develop case series for the further interpretation. Effective pharmacovigilance is dependent on availability of information in clinical effects. It requires a robust PV system, which includes a system of collecting and monitoring the ADRs and process for reviewing the data. Now I'll discuss about the adverse event case processing. These are the adverse event reporting forms: AEM, Adverse Event Monitoring Report; CEOMS, which is mainly used by the USA; MedWatch form used by the UK. Now, these are the steps of pharmacovigilance monitoring. First, the cases are registered, triage is done, registered, and then the cases are enrolled. Further, in the processing, what we do is all the data entry is done, all the information is put in the database, coding is done as per the MEDRA or the WHO dictionary, and then labeling, which I discussed in detail, the listedness assessment is done. Then further, the medical reviewers, the healthcare professional or physicians, they review the cases to serious cases, non-serious cases, and aggregate report review is done. And then further, the reporting is completed, assessment is completed, and it is recorded in the database. Now, when adverse drug reaction occurs, so what we do with that particular medicine? Either it is withdrawn, maybe temporarily or permanently. Either what the physician do, either it reduces the dosage or increases the dosage, or the dose is not changed depending on the adverse drug reaction. And if also it can be checked post therapy. Sometimes we are not aware of what action has been taken with the drug, we are not able to follow up with the patient, and in case of special situations, the action taken with the drug is always not applicable. That doesn't matter. Then comes the de-challenge and re-challenge. What is de-challenge? When we withdraw a drug and we record the outcome, if it is improved or not improved after stopping the drug, then we will come to know about the de-challenge. And what is re-challenge? Giving one drug again. Like we have temporarily withdrawn a drug and then we see the outcome of the adverse drug reaction, and then we again start that drug under the same conditions as before, and we record the outcome. If the event has reoccurred or if the event has not reoccurred, that is the re-challenge. So de-challenge, positive de-challenge, that is improvement of the reaction. Like when we have stopped giving our medicine, for example, a patient was experiencing headache with paracetamol, and we have stopped his paracetamol, and then we see if the patient is still having headache or if it has improved. So if it has improved, then we will say it is a positive de-challenge. Like resolution of a suspected ADR when the drug is withdrawn is a strong, although not conclusive indication of drug-induced reaction. Then comes the re-challenge. Re-challenge is positive when the symptoms reoccur on re-administration. When we have started the drug again and the event has reoccurred, so we will say it is a positive re-challenge. Negative re-challenge is failure of a symptom to reoccur. Like we have started the drug again, but it has not reoccurred. So it could be that adverse drug reaction could be because of some other possible reasons. So the re-challenge is negative. Next come the outcome. Outcome of the reaction or the adverse drug reaction or event at the time of last observation. So if you are receiving an initial report, at that time, the patient does not know, like the reporter, the person who is reporting, he is not aware of what is the outcome of the event. Then we will follow up, and in the next follow-up, maybe we will come to know that the outcome has recovered, that adverse drug reaction is in a recovering phase, or it is still not recovered, or it has recovered but some with complications, with sequel. So we'll get to know everything, and we may know that the patient has died. So that is that adverse drug reaction resulted in a fatal reaction. And outcome of subject when somebody is involved in clinical trials or some studies, so the outcome remains in the study, or it is withdrawn from the study, or the patient is lost to follow up, that is we cannot contact the patient again, or the death of the patient. Now I will discuss a case scenario with you. Like this way reporting is done. Like a 42-year-old female experienced vomiting during treatment with 200 mcg Pulmicort at night by inhalation for her asthma. The onset is on 3rd August until 5th, and she has been prescribed this drug since the end of July. The drug was stopped on 5th August and patient recovered. No re-challenge was performed and her doctor changed the drug. So now we have all the information We have the demographics of the patient, we have the age. We will add everything in the patient details in the database. Then we know what is the drug. It's the Pulmicort. We know the dosage of the drug. We know by which route it was taken. We know the therapeutic indication for that drug. Then further, we know the adverse drug reaction, the onset of the reaction, when it was cured, and when the drug was stopped, and if the rechallenge was performed or not. Everything we know, we will put all the details in the database in the different tabs available, and then this is how we can assess an adverse drug reaction with our drug. And then the labeling will be done, further the medical reviewing will be done, and this is how the case is recorded in the database. Similarly, the case 2. This is in this a 68-year-old male patient was started with Crestor on 29 January 2007 for hyperlipidemia, and on 2nd Feb the patient felt very ill. So this is the adverse event here. The patient felt very ill, and this is the start date, 2nd February. It started almost after 3-4 days, and upon admission, the diagnosis was myositis and abnormal hepatic function. Now the patient cannot tell you the diagnosis. When the patient was admitted in the hospital, some lab investigations were carried out, and the diagnosis was developed. That is myositis, and the hepatic function is also abnormal, that we can see from the ALT, which is 100 units per ml. The patient also took, now we will ask the patient for taking any concomitant drugs. The patient also took Sandimum oral for his bone marrow transplant rejection since October 2006. So Crestor was discontinued and the patient recovered a few days later. So if we have discontinued our suspect drug and the patient is recovering, so that means the rechallenge is positive. And they are mentioning already that no rechallenge has been performed. If they have not mentioned about the rechallenge, we can ask in the follow-up to the reporter. So reporter details must always be there. Identifiable reporter is a must in PV reporting, an identifiable patient, so that we can follow up with the case. If in initial all the information has not been reported, so we can follow up accordingly. There can be third party also which can follow up with the patient. There can be physicians also who are the reporters. They can follow up with the patient. This is another case in which a 67-year-old woman reported whose medical history included diabetes mellitus, hypertension, and has also been diagnosed with Alzheimer's. So in this case, in the earlier cases, medical history was not reported. We didn't know what were the underlying conditions of the patient. So here we know that the patient is already having diabetes, hypertension. So this can help us in the assessment of the causality and in the further assessment of the report. This woman been on long-term treatment of amlodipine, metformin, clapinclamide. She started receiving Exelon, Rivastigmine, 6 mg daily. After 5 months, she was hospitalized for dizziness and syncope. On admission, her BP was 90/60. These are the lab investigations which were carried out when the patient was admitted in the hospital. And this is a serious report because it led to the hospitalization of the patient. And also it's mentioned that women received a temporary transvenous pacemaker, and Exelon and amlodipine was discontinued. So this is amlodipine, the concomitant was discontinued here. Angiography was also done. After 3 days admission, her heart block spontaneously resolved and sinus rhythm was restored. Oral Exelon was restarted that day and complete heart block reoccurred. See, when our suspect drug was restarted, again the problem occurred which already cured. So it occurred on day 4. The woman received this permanent pacemaker the following day. Then further, due to the hospital stay, these drugs were stopped, and then it was restarted, and Exelon was also continued at the same dosage. So this will be put in the database accordingly in all the different tabs, and further assessment, listedness assessment, causality assessment will be done, which will lead to the final submissions of the case. Thank you. Any queries?
I think there is no question, ma'am. I think that
Okay.
the way in which you have covered, you have not only covered the basic, you have not only covered the case studies. I think entire aspects you have covered in your lecture. Both of you have covered all the aspects in your lecture. So whatever the query or doubt, if the participant will have, we will like to mail you with your permission.
Sure.
So thank you for this informative sessions. So on behalf of ACE, I would extend my deep gratitude to both of you, and thank you for sparing the time to give such a informative lecture on our platform. So
Thank you so much, sir. Thank you.
with this, we are
Thank you. Thank you.
with our session, today's session. We will join the next session and it would get declared later on very soon. Thank you very much, all of you. Now I am ending the broadcast. Thank you very much.
Thank you.
Thank you.